So we're moving on now to the last unit of this course, fourth unit, final unit of the course. This is the first lecture of material that'll be on exam 4. We're going to do the first half of chapter 17 today, pages 571 to 590, and chapter 17 focuses exclusively on the idea of the cytoskeleton. Today we're going to talk about only two components of the cytoskeleton. We'll start our discussion with the intermediate filaments. We'll talk about the role of intermediate filaments in structural support of the cell, as well as how they help support the nuclear envelope. We'll then move on to microtubules. We'll talk about microtubule polarity. There's two different ends or two different sides to a microtubule, and that plays a very important role in their function. We'll discuss how microtubules grow and assemble into filaments, and how microtubules are used to organize the interior of the cell. So cells do a lot of amazing things, and we've spent the first 3 units really highlighting some of the truly amazing things that cells do. But cells also do a lot of things that aren't so amazing, some more mundane, everyday things, such as change their shape, organize the location of their interior components. This includes vesicles, organelles, etc. The cells interact mechanically with their environment, retracting structures, protruding structures, etc. Cells carry out coordinated movements where a group of cells together will move in a coordinated way to achieve some larger multicellular response. And if you stop and think about it, even the more mundane everyday aspects and tasks of cells are in themselves pretty amazing. There's nothing a cell does that actually isn't amazing, so we won't lose our luster for all of these wonderful things that cells do. All of these things that I've listed here are mediated or carried out. They're possible because of the cytoskeleton of the cell. The textbook says the cytoskeleton is an intricate network of protein filaments that extends throughout the cytoplasm, and that's the best way to really describe this structure. It is a meshwork of proteins that permeates all aspects of a single cell. Here we see some fluorescent microscopy of the cytoskeleton, actin in red, intermediate filaments in green, and we also have the nucleus, the DNA in the nucleus stained blue. You can see that the cytoskeleton truly does permeate and encompass all aspects of the cell. Cytoskeletons are purely eukaryotic. There are cytoskeletal homologs, protein components in bacteria that are very similar to cytoskeletal proteins, but this entire network of protein filaments is exclusively eukaryotic. And even the term skeleton is a bit misleading, because the cytoskeleton is a very highly dynamic, constantly changing, constantly moving structure, unlike our own skeleton, which is quite static in its construction. So cytoskeletons are constantly changing, usually changing in response to signals that the cell is receiving. Because of this, the cytoskeleton really not only is the structural framework of the cell, the bones of the cell, the cytoskeleton is also the cell's muscles. The cytoskeleton is what allows the cell to move, as our muscles allow us to move. If a cell is moving, period, by moving we mean crawling along a surface like a fibroblast or an amoeba, we mean contracting like a muscle cell, we even mean dividing and differentiating into different morphologies. If a cell is moving, period, this cellular movement is possible due to the cytoskeleton. Without a cytoskeleton, cells could not move, period. Cellular mobility and motility would cease. In addition to this, cells, in their most basic comprehension, their most basic consideration, are nothing more than ATP factories. Cells make ATP, and the ATP allows the cells to survive so that it can make more ATP, so that it can continue to survive making more ATP. Really, in their simplest way, cells are nothing more than ATP-making factories, and like any good, efficient, well-designed factory, the interior of the cell is highly organized and highly compartmentalized. What allows that to be possible? The cytoskeleton. The cytoskeleton allows and controls the cellular localization of everything inside that cell. It serves as the major primary compartmentalization force, as well as the major thoroughfare for all cellular transport. You might remember those walking proteins we've talked about a number of times. Let's not forget, even for a moment, the other thing that we need the cytoskeleton for, mitosis and cytokinesis. Right here, we have captured the interior of a cell in the middle of mitosis. We have in green microtubules, those spindle microtubules you might remember from genetics class. In blue in the center are the chromosomes, the sister chromatids, highly compacted. And we have the attachment points in orange, where the microtubules are literally attaching to the centromeres, the kinetochores of the chromosomes. Chromosomes could not be pulled to opposite sides of soon-to-be daughter cells without microtubules, without the cytoskeleton. Cells could not divide. One cell could not pinch into two without the contractile forces of actin. And so the cytoskeleton is integrally involved in cellular division. Indeed, cells could not grow and divide. Cells could not reproduce without the cytoskeleton. So the cytoskeleton is hugely important for all cellular processes. We spoke briefly of the cytoskeleton before Exam 3. We had that very brief lecture about cellular components, and we included the cytoskeleton in that. Actually, excuse me, that was before Exam 2. My God, how time flies. But let's briefly review the brief basics of cytoskeleton before we move forward on the details. The cytoskeleton is built using 3 different types of protein filaments. Each of those protein filaments specialized for a different role of the cytoskeleton. We have the intermediate filaments, abbreviated IFs. We have the microtubules, abbreviated MTs. And we have the actin filaments, also called the microfilaments, and we'll just refer to these as actin from now on as well. So these are the 3 different types of protein filaments that come together and form the network, the protein network of the cytoskeleton. In this lecture, we'll talk about intermediate filaments and microtubules, and we'll save actin for our next lecture. So not only is each of these components different in its specialized role that it plays in the cytoskeleton, but each is also structurally different, made from different protein subunits as well. There is a whole slew of related, though different proteins that come together to make the intermediate filaments. Microtubules are made up of thousands, if not millions, of repeating monomers called tubulin protein. And, of course, the actin filaments are made up of actin protein. For each of these cases of cytoskeletal proteins, and for every single type of these filaments we're discussing, the proteins come together by the thousands, if not millions, to create very, very long fibers of protein complexes. These fibers, then, interweave and bundle with one another to create a wicker basket-like meshwork that is the cytoskeleton. So let's go through these very slowly across 2 lectures, one by one, and talk about how each of these filament proteins adds to and contributes to the cytoskeleton and what each of their role is. The specialty, the specialization of the intermediate filaments comes down to just brute force strength. The IFs are the strongest, most tensile of all the filaments that make up the cytoskeleton. So the main function of the intermediate filaments seems to be to help the cell withstand forces, withstand stretching forces, pulling forces, squeezing forces. This is all falls under the umbrella of mechanical stresses. So the cell is often the victim of all of these stresses, these mechanical stresses, and intermediate filaments allow them to survive those. Intermediate filaments are so named because their diameter is between the actin filaments, which are the smallest, and the microtubules, which are the biggest. But although their diameter might be intermediate, their toughness is the highest of all 3. So intermediate filaments are the most durable of the 3 different cytoskeletal components. In most cells, you find intermediate filaments in the cytoplasm. In the cytoplasm, intermediate filaments form their own network, which surrounds the nucleus, comes off of the nucleus, they're anchored at the nucleus, and the intermediate filaments extend outward to the cell membrane. And so they are the most pervasive of all 3 types of cytoskeletal components as well. You find them lining and attached to, anchored to the nucleus on one end, and then projecting out all the way to the cell membrane and anchored to the cell membrane on the other end. The intermediate filaments are truly found throughout the cytoplasm. Intermediate filaments are also found in the nucleus. This is a specific type of intermediate filament called the nuclear lamina. We'll talk about the nuclear lamina later on in this lecture. But the nuclear lamina is a type of nuclear cortex. Hopefully you might remember the cell cortex we've discussed before. the underlying protein meshwork or framework of the cell membrane. The nuclear lamina is an intermediate filament-based cortex or underlying structural support for the nuclear envelope. So you can see here is a diagram of the nucleus. This whole thing represents the nucleus. You see the nuclear pore complexes. We've talked about those. The nucleolus, here we mentioned that very briefly early on in the semester, is here in the center. And here is your nuclear lamina. There are your intermediate filaments lining the inside of the nucleus, providing structural support to the nuclear envelope, truly making the nucleus have a defined shape and structure. So, if intermediate filaments' main strength, if their specialty is strength, tensile strength, then it becomes important to consider how they're made and how their structure, how their synthesis contributes to their strength. Well, the best analogy for this is rope. Actually, intermediate filaments are made very much like rope. How is rope made? Well, rope is made up of thousands, if not millions, of individual fibers, which individually are actually quite fragile. But to make rope, these individual fibers are twisted together into strands. So you might see in this picture some of these fibers. See these little wispies here? These little wispies coming off the rope? Those are individual rope fibers. Now you see these bundles that are pretty easy to discern. These are twisted bundles of those strands. But that's not it. Now these strands are twisted together into larger bundles, and those bundles braided or twisted together into rope. This way of bundling and twisting strands provides an enormous amount of stretchable strength. It works for rope, and it works for intermediate filaments as well. We make intermediate filaments by starting off with an intermediate filament subunit, taking two of those and wrapping them together, truly twisting them together. Then these pairs, these dimers of intermediate filaments, are put together in a staggered orientation to create a tetramer, a group of four. These tetramers are bundled together, twisted together, to form the intermediate filament itself. Take a moment to compare these schematics. You see how this is a bundled network of individual strands? Just like this is a bundled and twisted network of individual strands. All rope has going for it is its tensile strength. And that's all intermediate filaments offer as well. Intermediate filament protein heads, that's what we see here in light blue, they're globular. They're not fibrous, they're not elongated, they're globular, they're spherical. And they are used to interact with many, many things. First and foremost, they are the anchoring points of the intermediate filaments. So the way that intermediate filaments stick to the nucleus, stick to the cell membrane, is through these globular heads. Intermediate filaments also stick to organelles, keep them from floating freely in the cytoplasm through these globular heads. Lots and lots of things allow intermediate filaments to interact with them through these globular heads. Okay, so that's how intermediate filaments are made. Now, what do intermediate filaments do? Well, we find intermediate filaments to be at their highest numbers, they're most abundant, in cells that experience the most mechanical stress. The axons of neurons are an example. These are very, very long, thin, fragile cellular structures that can be easily crushed or severed if not provided with some protein-based support. And this protein-based support comes from intermediate filaments. Muscle cells, thought of as very durable, but still just cells held together by very fragile cell membranes that are constantly contracting and relaxing, stretching and being stretched, require a great deal of tensile strength. Skin cells, and just press on your skin quite hard right now. No breakage, no tearing, no distortion, no pain. Those cells are able to be squeezed and pushed and pummeled and bruised. They don't break. That's due to the strength of intermediate filaments. Intermediate filaments do the same thing in all of these cell types, provide tensile strength. They stretch. They stretch, and by stretching, they distribute the forces of that mechanical stress, of that squeezing, pushing, pulling. They distribute those forces throughout the entire cell, rather than those forces being localized to the one point of stress. By providing these intermediate filaments and the strength that they provide, cells can be stretched and distorted, but remain intact and remain connected to one another. Without that durability, those same cells would stretch and tear and separate, and you would have cellular destruction. So without intermediate filaments, cells could not withstand the forces that are put on them. The cells could not distribute these forces and spread them out over the tissue, resulting in those tissues tearing. This is not novel. This is not a novel idea to us. Like many of the things that cells have been doing for thousands, if not millions of years, we stole this idea before even realizing that cells were doing it first. The whole idea of carbon fibers in fiberglass are these linear, stretchy, tensile strength polymers providing greater strength to what would otherwise be a fragile structure. The whole reason we put steel rods in reinforced concrete is to put tensile strength in what would otherwise be a fragile, breakable, crackable slab of concrete. By putting fibers with tensile strength into more fragile structures, whether it be fiberglass, bridge ties, or cells, we add to the tensile strength in the distribution of forces. We add to the mechanical stress that those structures can endure before breaking. We put linear polymers in our materials to achieve a resistance to tearing and breaking under mechanical stress, and the cells do the same exact thing with intermediate filaments. There are four main classes of intermediate filaments that we'll discuss and differentiate between. The first are the keratins. The keratin filaments can be found in all epithelial cells. These are cells of the digestive lining as well as the skin. These intermediate filaments, the keratins, they are the broadest type found in the cytoplasm of almost every cell of every vertebrate animal's epithelium. There are specialized keratins that also make up feathers, claws, fingernails, hair. This is where we're more familiar with keratin. Each keratin intermediate filament is made up of a mixture of different but related subunits. This makes keratin the most diverse class of intermediate filaments as well, because you can mix and match a lot of those individual strands. Remember individual strands dimerized, they wrapped around each other? And then two dimers stacked in a staggered orientation to make a tetramer? For keratins, there are lots and lots of different individual monomers that can twist together into dimers and tetramers, allowing a very large variety of different keratin intermediate filaments to be made. Like all intermediate filaments, keratins distribute the stress of stretching, especially in epithelial cells, like the skin and like the gut. There's a disorder in humans called, and I'll do my best here, epidermolysis bullosa simplex. This is a genetic disorder. It stems from a mutation in keratin protein genes, so in some of the genes that encode the keratin proteins. And this mutation results in the keratin proteins not bundling properly. What are the effects of this syndrome? Well, as a result of this mutation, the skin is very vulnerable to mechanical injury. Even very gentle pressure on the skin will cause severe tissue damage and bruising due to the lack of proper bundling of the keratins. I tried to find a picture of these for you, but to be quite honest, all of the pictures were a little bit too disturbing. The severity of these injuries due to very, very mundane everyday tasks where I was not comfortable putting them on the slides. And so it really highlights the importance of the keratins in general, specifically, and the intermediate filaments in general. I brought it up just a second ago by accident, but it's also worth pointing out the ridiculous, bogus scams that are out there. So this is a very, very expensive conditioner, I guess it is, some hair finisher that's called Keratin Complex. And this conditioner is supposed to smooth your hair, make your frizzy hair look as beautiful as the hair of the model on the bottom of this panel. Please keep in mind, this is just nonsense. Keratin is nothing more than a filament protein that your hair is made of. Adding keratin to your hair does not help your hair's health at all. The keratin in this conditioner is washed away down the drain like everything else we rinse out of our hair. What's adding to the apparent health of the hair on the model on the bottom is nothing more than whatever smoothers and conditioners that they're putting into these this keratin complex product anyway. So whenever you see protein added to your shampoo, protein added to your conditioner, that's just nonsense. That protein's not helping in any way. All right, so the second class of intermediate filaments, the vimentins. The vimentins and their related intermediate filaments that are simply called vimentin-related filaments are found in connective tissue. So things like cartilage and the pericardium tissues of our torso, of our central cavity. They're also found in the glial cells, the support cells of the nervous system. In all connective tissues that are involved in tissue-to-tissue or organ-to-organ connections, we find the vimentins. They are extremely durable and allow a great deal of stretchiness, a great deal of tolerance to mechanical stress. The third class are the neurofilaments. Pretty self-explanatory. It's clear where you find these, but the neurofilaments are exclusively found in the nervous system. They are the proteins that provide the axonal structural support. They are especially important in long, thin, fragile axons, the axons of the central nervous system, especially those that project from the base of the spinal column to the legs. It's interesting, but they also seem to play some role, either directly or indirectly, with neural regeneration. The cells of the peripheral nervous system have a different type of neurofilament makeup than the cells of the central nervous system, and that seems to somehow allow regeneration in the peripheral nervous system, where, of course, we know that there's no regeneration of neurons broken in the central nervous system. This is why paraplegics never heal. The fourth class we've already mentioned in this lecture, but they are the nuclear lamins. The nuclear lamins are intermediate filaments that are involved in providing strength and structural durability to the nuclear envelope of all animal cells. And so let's be clear here that we have 4 classes total. 3 of them are cytoplasmic. The keratins, the vimentins, and the neurofilaments are all found in the cytoplasm of their respective cells. Epithelial cells for the keratins, connective tissue cells for the vimentins, and neurons for the neurofilaments. The nuclear lamins, our 4th class, are in all types of animal cells, but they are exclusively nuclear. So let's talk a little bit more about this nuclear lamina. So we've got some cytoplasmic classes for intermediate filaments, excuse me, but we only have one nuclear class for this protein type, and those are the lamins. The first 3 classes that we talked about are also rope-like. They are made in the way that we've already described. The intermediate filament subunits are bundled into dimers and then bundled further into tetramers and bundled further into rope-like long filaments that provide tensile strength. The lamins are different from that. The lamins form a 2-dimensional mesh, truly the wicker basket idea. The laminins run perpendicular to one another, so these are individual lamin bundles we see here, running perpendicular to each other, being interwoven with one another, truly forming a 2-dimensional protein mesh. In addition, unlike other intermediate filaments, lamins are extremely dynamic, and they actually have to be. See, the lamins provide the structural framework for the nuclear envelope. But you all know from your genetics classes that with every single round of cellular division, the nucleus disassembles. The entire nucleus dissolves right near metaphase. The entire nucleus dissolves, and then after cytokinesis, the first step of post-cytokinesis prophase is that the nuclear envelopes for the new daughter cells reforms. Every time the nucleus dissolves at the beginning of mitosis and it reforms at the end of mitosis, we also must have a dismantling of the nuclear lamins, and then a reassembly of the nuclear lamins for a reassembly of the nuclear envelope. So lamins must be extremely dynamic, because they must disassemble and reform with each round of mitosis. I don't want to bother anybody here by bringing up mitosis, but we will have to revisit the idea of mitosis, not only now, but as we move further into the cell cycle lectures, which come after this chapter. So unlike all the other classes of the intermediate filaments that are in the cytoplasm, nuclear lamins have to be extremely dynamic. They cannot be permanent. So how is this dynamic nature of the lamins controlled? Well, it's controlled in a way that is not foreign to us, phosphorylation. There are kinases which specifically phosphorylate the lamins, and there are phosphatases for laminin dephosphorylation. More specifically, When we phosphorylate lamins using a kinase, we weaken their affinity for other lamins. We create a type of repulsive property where lamins do not want to bind with other lamins, and this promotes disassembly. When time comes to reassemble the lamin, reassemble the nuclear envelope, a phosphatase dephosphorylates those lamin proteins, promoting assembly back into the meshwork and allowing the nuclear envelope to reform. Very, very highly dynamic process, very interesting process. So that's one way of stabilizing intermediate filaments, by dephosphorylation. If we phosphorylate intermediate filaments, we cause them to lose their affinity for one another. But intermediate filaments are also stabilized and reinforced by binding to accessory proteins. We'll have just one example of just such an accessory protein, but it is only an example. There are many, many other cases where this is true, but we'll use the example of plectin. Plectin is a protein that binds to intermediate filaments and, through binding, stabilizes them. Plectin, as well as other accessory proteins, binds across intermediate filament bundles, cross-linking them into stronger two-dimensional arrays. So in this diagram here, in red are our intermediate filaments, and notice that they're running parallel to one another. Now they're made like rope, so they're providing a great deal of tensile strength, but they would be even stronger if they were bundled. The plectin in light blue is what's bundling these intermediate filaments, making them more durable, more strength. Proteins such as plectin also link intermediate filaments to other components of the cytoskeleton, providing even greater strength against mechanical stress. So now we can cross-link intermediate filaments to microtubules or to actin, creating a true one single interwoven cytoskeleton. These accessory proteins are extremely important in that regard. Also highlighting the importance of proteins such as plectin are mutations in them. Remember how we could see how important intermediate filaments were by looking at the epidermolysis bullosa. So what happens if we mutate something such as plectin? A mutation in the gene for plectin causes an even more devastating skin disorder. The skin disorder caused by the mutation in plectin takes the worst of the symptoms of epidermolysis bullosa simplex, but combines them with muscular dystrophy as well as neurodegeneration. In fact, knockout mice made without plectin die. They're not even viable. So why would that be? Why would a mutation to plectin, not even a cytoskeleton protein in its own right, cause a disease or a syndrome that is even worse than mutation to an intermediate filament specifically? Well, remember, epidermolysis bullosa was specific for the keratins. So we didn't affect neurofilaments and we didn't affect the vimentins. Plectin is involved in cross-linking and stabilizing all classes of intermediate filaments. So we get the effects of keratin mutations. We also get muscular dystrophy and neurodegeneration due to breakdown of neurological systems. And so by affecting the cross-linking and bundling of the cytoskeletal components, we actually have a disease that's even worse than mutations to the cytoskeletal components themselves. So that's it for the intermediate filaments. Pretty straightforward story. Simple structural support, nothing more than that, providing tensile strength to the cell so that it can withstand all of the different mechanical stresses that a cell has to endure. We'll move on now to the two more interesting stories, the microtubules, which will be used to wrap up the rest of this lecture, as well as the actin filaments and how the actin filaments allow cellular motion, which we'll talk about in the next lecture. So let's move on to microtubules now. Microtubules play a very crucial and important role in organizing the interior of eukaryotic cells. Microtubules are very long and stiff tubes of protein. Now that's important. They are hollow on the inside. That's not the case for intermediate filaments. Intermediate filaments were rope. Microtubules truly are tubes. They are hollow. And they are tubes of protein that can be rapidly disassembled from one location and reassembled elsewhere. So they have a dynamic component. Unlike intermediate filaments, microtubules must originate from specific cellular structures. We'll talk about what those structures are in just a moment. But intermediate filaments could potentially form anywhere. Yes, they have to be anchored on both ends, but they can form anywhere. Not the case for microtubules. Microtubules come from one place and one place only. And we see that here. In green, we see the origin points of microtubules, and the microtubules are these green threads coming off of those. This, again, is a cell caught in the middle of mitosis, so you might see it in the center here, right, where the arrow is pointing. There are blue chromosomes. These chromosomes are going to be pulled apart. But these microtubules all have a common point of origin. That's not true for intermediate filaments. Microtubules grow out of and originate from very small structures, actually, near the center of the cell, close to the nucleus, called the centrosome. And again, the centrosome comes up very briefly in mitosis because the centrosome is where the microtubules come from. But all microtubules come from centrosomes, even those not involved in mitosis. This is just a really cool picture of a centrosome, a general artist's depiction of it. But you can see the centrosome almost looks like a spoked ball. All of these tubes coming out of this central structure here are microtubules. They all originate at the centrosome. So we have to talk about it again. Another mitosis alert here. Once the microtubules project out of that centrosome, they're going to anchor. They can anchor at the interior face of the cell membrane, thereby creating protein tracks throughout the entire cell. Our walking proteins, which we've talked about before, walk along these tracks to move cargo around the cell. We also talked about transport vesicles being transported with clathrin-coated pits and Rab proteins and tethering proteins. We mentioned that transport vesicles are moved along these microtubule tracks. Those microtubule tracks is what we're discussing now. Microtubules also anchor and localize organelles, keep them from floating away. Yes, intermediate filaments did that, but microtubules do them as well. And unlike intermediate filaments, microtubules are much, much more about function than structure. The true underlying structural strength and support of the cytoskeleton, that is the job of the intermediate filaments. However, the cytoskeleton also has the job of providing the thoroughfare, the major highways throughout the cell for getting things shipped from one place in the cell to the other. That is the specialty of the microtubules. The microtubules are the cables and the roads by which things move around the cell. And of course, I guaranteed you that mitosis alert. Don't forget that microtubules are the very same structures that reach out, bind to sister chromatids, and pull them apart during anaphase of mitosis. So we have our centrosome, the point of origin of our microtubules, and the microtubules come out of that centrosome. For mitosis, the cytoplasmic microtubules are actually disassembled, completely taken down. All of those tracks for cellular transport are removed, and then those tubulin subunits, the building blocks of microtubules, are reassembled into new microtubules that make up the mitotic spindle. The mitotic spindle sends microtubules out to those chromosomes and pulls apart the sister chromatids during mitosis. Not all microtubule-based structures are so dynamic. Some are quite static. Microtubules are the protein basis for the motile structures of motile cells. So flagella are microtubule-based. Cilia are microtubule-based. These are microtubules that are never taken down, even for mitosis, and they are motor microtubules, microtubules for cellular movement. These structures and the microtubules that make them protrude or extend out from the surface of the eukaryotic cells that house them. I already mentioned it, but microtubules are made up of a subunit of individual monomers called tubulin proteins. Tubulins are proteins that themselves dimerize, so two tubulins come together to form a globular dimer, such as what's shown here. Each of those dimers has an alpha subunit and a beta subunit. Maybe you're already wondering, well, those look like balls of protein. And how in the world is a ball of protein going to make up a fiber? How do you make a fiber protein of microtubules out of spherical proteins such as these alpha and beta subunits of tubulin? Well, good question. Dimers stack together. They stack together by interacting with non-covalent forces, H-bonds, hydrophilic interactions, etc., etc. And by doing so, they create a long chain of globular spheres. Now this long chain of globular spheres alternates alpha subunit, beta subunit, alpha subunit, beta subunit, alpha subunit, beta subunit. And one of these creates one portion of the wall of the hollow microtubule. Now imagine 13 of these alternating chains of alpha and beta tubulin subunits, 13 interacting side by side, not end to end, making a hollow tube of protein. So now you've got not one, but 13 of them. 13 of these chains interacting side by side creates a hollow tube of protein. I already said, but each of these dimers is made up of two different subunits, an alpha subunit and a beta subunit. Because these subunits are alternating, alpha, beta, alpha, beta, alpha, beta, and because they are all oriented in the same direction, microtubules have a beginning and they have an end. They have polarity. Each protofilament meaning one of these individual chains, one of these 13 that makes up the tube, faces in the same direction, is oriented with the same pattern. You see the way they're lined up here, they're all oriented in the same way. Because of that, all of the beta subunits are localized at one end, making a ring of beta subunits on one end, and on the other end there is a ring of terminating alpha subunits. This is the polarity. The ends of microtubules are referred to not as alpha and beta, but as plus and minus, just for convenience. And indeed, just to show you that this truly is the structure of microtubules, these are electron micrographs. True pictures taken from the electron microscope of the side of a microtubule. You can see the protofilaments. Even in the electron micrograph, you can see the individual protofilaments. You can also pretty much discern the individual tubulins. And then that same microtubule, cross-section cut in half, and you're looking down the barrel, just as you are in this schematic here. And here you can easily see the individual tubulin proteins and the hollow interior. Microtubules, of course, form in vivo, form in living cells, and as they form, tubulin dimers are added to both ends of the microtubule. So tubulins can be added to either the plus or the minus end of a growing microtubule fiber. However, they are added more efficiently to the plus end. So the plus end is so named because it is the growing end of the microtubule. It is where tubulin dimers more easily join the growing fiber. The polarity of microtubules becomes important for their function. Remember, microtubules' specialty is not strength, but transport. And when you're going somewhere, when you're getting somewhere, where you're moving, direction, polarity is important. When you consider that the specialty of microtubules is cellular transport, it becomes a necessity that the two ends have to be physically and chemically different. The microtubules are the highways and the byways of the cell. They are the roads by which the walking proteins walk to move things along in the cell. These structures are the shipping lanes for all cellular transport processes. So just as roads have one direction, the right lane to go east and the left lane to go west, the right lane to go north and the left lane to go south, microtubules also must have their directions delineated for transport purposes. Individual walking proteins walk on microtubules depending on this polarity. Some proteins walk towards the minus end, and some proteins walk towards the plus end, and this becomes crucially important for trafficking of vesicles and other cargo throughout the cell. The textbook says if microtubules had no polarity, they could not serve their function in defining a direction for intracellular transport. It is this minus and plus end. It is this polarity that allows proteins to know in which direction they are going in order for shipping proteins. And we'll come back to that near the end of the lecture. Let's first get back to this incredible idea of the centrosome and why centrosomes are needed as a point of origin for forming microtubule bundles, microtubule fibers. And start that conversation by just asking, what the hell is a centrosome? Well, microtubules form rapidly, and they form efficiently, especially when tubulin dimers are being added to a pre-existing microtubule fiber. However, tubulins don't really like being fibers. And so, when trying to start a microtubule from scratch, it's a very, very hard thing to do. Tubulins will join an already existing microtubule fiber quite readily, but tubulins won't assemble into that fiber on their own. So all centrosomes really do is serve as tubulin organizers. Centrosomes coax initial tubulin dimers into forming that protofilament, into forming that initial short microtubule. Once that microtubule has been formed, then additional tubulin dimers can be added to it, extending it and making it longer. But that first initializing event of making that first tube is what centrosomes make possible. Once that tube has been started, more tubulin will join it quite easily, turning a short tube into a long tube. But centrosomes are needed to start that party and get that process started. So how do centrosomes do that? Well, centrosomes are, again, a ball of structure, to keep it as simple as possible, containing hundreds of ring-shaped structures made up of tubulin. Here it's a third type of tubulin, gamma tubulin, if you're so curious. They're not alpha or beta, it's a third type, but still tubulin. So these are rings of tubulin protein. These rings, because they are tubulin, serve as the smallest, shortest, pre-existing tubes of tubulin. They serve as nucleation sites, as starting points for microtubule formation. Alpha and beta dimers of tubulin will join onto these rings, just as they would join pre-existing tubes. And as those alpha and beta subunits join, they extend the length of the microtubule, starting at the ring on the centrosome and extending outward. The minus end of the microtubule, again keeping this idea of polarity in mind, is the end that's embedded into the centrosome. The plus end is the growing end, and it grows outward from the centrosome towards the cell periphery, meaning the interior face of the cell membrane. Once a microtubule has been started at a centrosome, it grows. Tubulin dimers are added to the plus end grows out, and eventually we make contact with some other structure. And this growing, this addition of new alpha and beta dimers being added to a growing microtubule, this lasts for minutes. Sometimes, though, quite often, without warning, a very peculiar thing happens. Just as quickly as it had started to form, that same microtubule begins to disintegrate, and it unravels, and it retracts back towards the centrosome. In other words, the dimers start falling off of the plus end, causing the microtubule to shorten. So while some microtubules are growing outward away from the centrosome, other microtubules are retracting back towards it. As dimers fall off of that plus end, the microtubule shrinks. Sometimes it falls apart partially, and then it continues to grow again. Sometimes it falls apart entirely, all the way back to the gamma ring of tubulin at the centrosome itself. This whole process is called dynamic instability, the constantly changing and unstable nature of microtubules. And it's crazy. It is incredibly energetically costly to make microtubules, yet just as often as them being made, they collapse and fall apart again. Why? Well, it comes down to energy and affinity. Each alpha-beta dimer of tubulin is capable of binding either GTP or GDP. Yep, here we go again. GTP binding dimers, that is, alpha-beta dimers of tubulin that are bound to GTP, pack efficiently and readily into microtubule fibers. However, those same dimers, when binding to GDP, don't have a high affinity for assembling into microtubules. Usually this is okay. When microtubules are being rapidly polymerized, the dimers that are being added onto the end of the fiber are containing GTP, and so they form a stable cap because the GTP containing dimers pack efficiently and readily into microtubules. The alpha-beta dimers deeper into the fiber have hydrolyzed their GTP to GDP, so they are no longer stable microtubule subunits, but they're so integrated into the fiber already that they can't pop out on their own. So what we have in this case is what we call a stable GTP cap of the microtubule, meaning that all of the alpha-beta dimers at the end of the growing microtubule contain GTP, because that microtubule is growing so fast, and that's a stable cap holding the rest of the microtubules in place, even though they contain GDP. But when microtubule polymerization slows down, and it slows down to the point where the GTP in those tubulin dimers at the cap have also hydrolyzed their GTP to GDP, now we have unstable, low-affinity dimers at the cap, and the end of the microtubule frays. The GDP-containing tubulin dimers at the cap are not stably there, and that's the weakest point of the microtubule, so the microtubule begins to fall apart at the cap. This preponderance of GDP-containing microtubule dimers at the cap tips the balance towards the disassembly of the fiber. Remember, the rest of the fiber contains GDP. That was the unstable portion that the cap was keeping static. But now, as that cap begins to fray, the rest of the microtubules also begin to fall apart. The rest of the tubulin dimers also become unstable. And so this becomes an avalanche of instability. Once disassembly starts, it often doesn't stop because all of the tubulin dimers contain GDP, and all of them technically don't want to be there. The frayed end of a microtubule fiber becomes a frayed fiber entirely. Once microtubules disassemble, those tubulin dimers are freed. They join the pool of all the other free tubulin dimers, and they wait to release their GDP, bind GTP, and once again enter the polymerization stage. All of this that we just described, this idea of dynamic instability, is quite different from intermediate filaments. The main feature of intermediate filaments was strength and stability. Intermediate filaments don't spontaneously unravel. If you want to disassemble an intermediate filament, you have to actively go ahead and do that. Intermediate filaments are made of fibrous proteins. They are fibrous in their very nature. Microtubules, on the other hand, are globular, and so they more easily disassemble back to their globular subunits. Intermediate filaments are remarkably stable proteins, whereas microtubules, dynamically unstable. And finally, intermediate filaments are purely structural. Their only point is to provide structural stability, whereas microtubules are the cabling and roadways of the cell, providing polarity for transport. So, although they are both cytoskeletal components and they both contribute to the overall structure of the cell, take a moment to appreciate the striking differences between these two components of the cytoskeleton, and make special note how these differences give them their specialized roles in the cytoskeleton. Alright, so let's go back to this idea of dynamic instability and why you would ever do this as a cell and waste this energy. So, if microtubules are so important, why do they rapidly fall apart all on their own? And more importantly, how do we stop this from happening? Well, we stop microtubules from disassembling by anchoring the plus growing end to something. Once the plus end of a microtubule is anchored to something, is linked to something, now the minus end is going to be embedded into the centrosome. That'll never change. It's the plus end that needs to find a stable place to anchor. But once the microtubule is anchored in two places, that microtubule is stable. And it will continue to be there until it is actively disassembled. So now we get to the idea of dynamic. Why are microtubules dynamic? The dynamic nature of microtubules is actually quite ingenious. It is a waste of energy, but it's the only way to accomplish a special feat. See, the cell has no way to target microtubules to specific locations. That's a function that just doesn't exist in the cell. If the cell wants to send a microtubule from the centrosome to the cell membrane, it can't do that intentionally. It can merely start making a microtubule. So the centrosome is constantly sending out new microtubules in all directions at all times. Those microtubules that catch on to a good target become stably associated with that target. They anchor to that target on their plus end. The minus end remains embedded into the centrosome, and again, if both ends are anchored, the microtubule is stable. If a microtubule is sent out of the centrosome and it doesn't find a suitable place to anchor, doesn't find the right place to anchor, it is retracted. So microtubules that are sent out and miss an appropriate target, for lack of a better word, disassemble back to the centrosome. And so this figure shows us that we have our centrosome sending out microtubules in all directions. Some of those microtubules don't hit anything of any consequence, and they retract back to the centrosome. Others, however, do land on an appropriate docking or anchoring point, and those microtubules do not retract. The plus end stably anchors there, and we eventually, in this kind of random, blindfolded way of sending out these lines, anchor the microtubules to all the structures that we need anchoring to. It's very much like fishing. You throw out the line randomly, having no idea if you're going to catch the anchoring point you wish to catch. When you do catch that point, that's where you stably keep your line in. If you throw the fishing line into the water and it doesn't find a point to anchor, what do? You retract it all the way to the beginning and you cast out a new line. Try to find a new place to anchor. So microtubules are really just the centrosome fishing for good places for microtubules to attach. When those places are found, you have a stable attachment. When those destinations are missed, the microtubule retracts. Again, as random as it sounds, as crazy as it sounds, as wasteful as it sounds, this strategy allows a cell to set up a highly organized, highly interconnecting system of microtubule fibers. And we see that this process is required and necessary because drugs that block the stabilization of microtubules block mitosis. In fact, such drugs that block mitosis by interfering with microtubule stabilization have been fairly effective anti-cancer drugs because by blocking mitosis, they block cell division, and by blocking cell division, they block cancer progression. Some examples of such drugs are Taxol, colchicine, and vinblastine. I'm not sure I'm pronouncing that one correctly. I've never heard it pronounced before. They act in similar ways, but slightly different. Taxol binds to and stabilizes microtubules, stops them from being disassembled. If all the microtubules you made are the only ones you're allowed to make, you can't disassemble them in order to make mitotic spindles. Colchicine blocks subunits and prevents the polymerization of microtubules into new microtubules, as does the second drug, vinblastine. So now we've established our microtubule network. We've established our system of transport using microtubules. Let's talk briefly and wrap this lecture up by talking a little bit about our walking proteins in some detail. Like microtubules themselves, many cells have their own polarity. They have two distinct ends. Neurons, of course, are our most extreme example of cellular polarity, but the same is true for circulatory cells, cells of the capillaries, etc. For neurons, we have a cell body at one end, we have an axon terminal at the other, and the two ends of the cell are extremely different and specialized for the cellular function, and oftentimes the polarity of the cell is reflected or mirrored by the polarity of the microtubules. For the neuron, that's the case that the plus end of microtubules are found in the axon terminal, the minus end of the microtubules are found in the cell soma, in the cell body. This polarity helps to guide vesicle transport and keep vesicles going in the proper direction. And this polarity of microtubules also serves to position and segregate organelles appropriately. We talked about the walking proteins before, but we never really officially covered them. We'll do that now. First of all, let's not call them the walking proteins anymore. That's more of my own little pet name for them because that is how they move. But walking proteins are collectively referred to as motor proteins. The motor proteins include myosin, the muscle proteins, which we'll discuss in our next lecture, probably the most amazing of all the motor proteins, but also they include the walking proteins of the microtubules. The motor proteins all use energy from ATP hydrolysis in order to walk. In the case of microtubules, these proteins use ATP hydrolysis to travel along those microtubules in a single predefined direction. Again, to give us some lay of the land here, what we see is a beautiful picture of a microtubule here running along the bottom. This seems to be a vesicle, transport vesicle, that's being carried. The transport vesicle is held to the motor protein through these two binding domains of the motor protein. And here at the other end is the region of the motor protein that is doing the walking. These regions hydrolyze ATP and walk along or adhere along the microtubules in a single direction, dragging the cargo vesicle along with it. So all of these motor proteins have the ability to be attached to cargo, often cargo that's loaded into transport vesicles, on one end, and on the other end move along the cytoskeletal component, facilitating transport. Dozens of different motor proteins have already been identified. They differ in the cytoskeletal component that they can bind to and walk along, in the direction that they move in. So do they walk towards the positive end, or do they walk towards the negative end? And also differ in the cargo that they can carry. Do they carry specific proteins? Do they carry vesicles? Do they carry vesicles only from specific organelles? Etc. The two motor proteins we'll end this lecture with are kinesin and dynein. Kinesin and dynein are both motor proteins that travel along microtubules. The main difference between them is that kinesin walks towards the plus end of the microtubule, while dynein walks towards the minus end. We could say this another way, though. Kinesins move away from the centrosome, where dyneins walk towards the centrosome. Both of these proteins exist as dimers. Both of them bind ATP. Both of them cleave ATP to provide the energy for walking. In each of these cases, the head domain is defined as the region of the protein which actually binds to the cytoskeletal filament. This throws students for a loop sometimes, because that's the walking part. What seems to be the feet of the protein actually is called the head domain. These domains also have the ATPase activity. The tails of the protein are those that bind to the cargo. So I'm going to back up for a second. But these are the head regions of these walking proteins, and this is the tail of the protein in green, directly bound to the cargo being carried. We'll talk a little bit more about the cycle of motor proteins in our myosin lecture and the actin lecture that we're going to cover next, but suffice it to say now very briefly, there is a cycle of binding to ATP, cleaving that ATP, and then releasing the resultant ADP that results in the conformational changes in these motor proteins. When these motor proteins bind ATP, oftentimes they let go of their cytoskeletal components. So in the case of dynein and kinesin, when ATP is bound, the head region lets go of the microtubule. When the ATP is cleaved, we take a step forward on the microtubule. And when the ADP is released, we actually rebind to the microtubule. So again, we have the binding of ATP for release of one of these head domains, cleaving of ATP to take a step forward on this head domain, and then releasing ADP to lock down this head domain, while the other binds ATP. So this head domain just bound ATP, releasing it from the microtubule. It will cleave that ATP and swing forward, taking another step ahead of this head domain. And then by releasing ADP, this will lock down onto the microtubule here. This is how these walking proteins walk. It's quite incredible. Organelles also move along microtubules. Before I get to that, though, I do want to harken back to the idea of the polarity of the neuron. Please keep in mind, as we said, the plus end of the microtubules points towards the axon terminal, where the minus end of microtubules are embedded in the soma. So if you had a shipment of neurotransmitters, let's say, that you had just produced in the cell body, and you wanted to ship those neurotransmitters to the axon terminal, you would load them into a vesicle and load that vesicle onto kinesin, because kinesin walks from the soma to the axon terminal. However, if you had some material in the axon terminal that you wanted to get into a lysosome in the soma, you would load that material into a vesicle at the axon terminal and load that vesicle onto dynein. And dynein would walk towards the minus end, towards the soma of the cell, where the lysosomes were, transporting your cargo there. So depending on which direction in the cell you want to move in, you would ride a different walking protein. This is how the polarity of the microtubules figures directly into the cellular transport, because these walking proteins, these motor proteins, walk along specific microtubules in specific directions. So let's wrap up talking a little bit about organelle movement along microtubules. Both the endoplasmic reticulum as well as the Golgi are positioned in the cell. They have their cellular locations due to their interactions with microtubules. Kinesins, remember kinesins attach to the cytosolic face of the ER, and dyneins attach to the Golgi. What that means is that once attached attached, these proteins respectively walk along microtubules as the cell develops and grows outward, stretching out their respective organelles. I can analogize this to the spreading of a tarp over a baseball field when it starts to rain. Of course, this is Yankee Stadium, the best stadium for the best team in all of the major leagues. But when it's time to cover the field, you get your walking proteins, the people, who grab onto the edge of the membrane for these organelles, grab onto them and walk along the field, spreading that membrane out. The membrane proteins, the motor proteins, are binding to the membranes of these organelles and walking along microtubules, spreading those out. When cells are treated with chemicals that cause the disassembly of microtubules, what we see is that the location of organelles such as the ER as well as the Golgi change dramatically. The ER literally collapses or shrinks towards the center of the cell. Well, why might that be? See, the kinesins were attached to the ER, stretching and walking the ER membrane away from the nucleus. So when microtubules collapse, the ER membrane springs back towards the nucleus because it's no longer being stretched out. The Golgi fragments into small vesicles, which disperse throughout the cell. Why might that be? Well, the vesicles, the membrane of the Golgi were being held all in one spot by those vesicles, those membranes, being held to the cytoskeleton through dynein, and dynein kept those membranes all together. As microtubules disassemble, the dyneins have nothing to walk on, and then the membranes of the Golgi fragment and disperse. Interestingly, amazingly, as soon as that chemical that disrupted microtubule assembly is removed, microtubules spontaneously reassemble. The motor proteins which remained associated with the membranes of these organelles latch back onto microtubules, they begin to walk again, and they automatically reposition the organelles to where they should be. Amazing, amazing that the spatial organization of the cell is pre-programmed in these motor proteins as well as the microtubules that they walk on. So I hope that even already I've convinced you that the cytoskeleton is far, far more than the simple scaffolding of the cell. It is highly functional, highly dynamic, and critical to all cellular processes. So, what did we talk about today? Well, we started off with the basics. The cytoskeleton in its totality is essentially an intricate network of protein filaments that come together, forming an extensive network throughout the cell, throughout the cytoplasm. The cytoskeleton is structural. Yes, it is the scaffolding of the cell. It is the skeleton of the cell, but that's not all it is. It is also the cell's muscles. The cell can move and change its shape due to the dynamic nature of the cytoskeleton. The cytoskeleton is built using 3 different main types of fibrous proteins that we refer to as the intermediate filaments, the microtubules, and the actin filaments. The specialty of the intermediate filaments is pure tensile strength. They are built like rope. They help the cell withstand mechanical stresses such as pulling, squeezing, pushing, etc. There are 4 main classes of intermediate filaments: the keratins, which are found in epithelial cells, the vimentins, which are found in connective tissues, the neurofilaments, self-explanatory intermediate filaments of the nervous system, as well as the nuclear lamins, which line and support the inside of the nuclear envelope. All of these intermediate filaments are made in the same, the first 3 classes are made in the same way, where dimers of intermediate filaments bundle. They then form tetramers. Tetramers come together to form long filaments, such as rope. The nuclear lamins form a 2-dimensional meshwork of protein, which provides a wicker-like, intermeshed, interwoven-like support for the nuclear envelope. Then we moved on to microtubules. Unlike intermediate filaments, microtubules have a single point of origin called the centrosome. All microtubules are made up of subunit proteins called tubulins, alpha and beta subunits respectively. Because these alpha and beta subunits are oriented in the same direction, each making a linear protofilament, 13 protofilaments coming together to form a hollow tube of tubulin, because they are aligned in the same way, microtubules have polarity. They have a beginning and an end, a plus end and a minus end. This polarity figures integrally into the transport function of microtubules. In fact, if microtubules had no polarity, they could not serve any function in defining the direction for intracellular transport, because we have motor proteins which walk along microtubules in the direction that their polarity points. Microtubules are made using the principles of dynamic instability. The centrosome casts out a line of microtubule, hoping that randomly it will collide with and anchor to a suitable location for that microtubule to be interacting with. If no suitable anchoring point is found, that microtubule retracts. It retracts because of the too slowed polymerization of tubulin on the growing end. Fast-growing microtubules have a GTP cap, a stable GTP cap, which allows that microtubule to remain growing. If the formation or polymerization of a microtubule slows, the GTP cap is lost because the GTP is hydrolyzed to GDP. The cap frays, and then the rest of the microtubule frays, resulting in retraction. When the plus growing end of a microtubule finds a suitable anchoring point, it anchors there, and this stabilizes the microtubule. We ended our discussion with the motor proteins. Kinesin and dynein were our examples. Kinesin walks towards the plus end, away from the centrosome. Dyneins walk to the minus end of the microtubule, towards the centrosome and the nucleus, again highlighting the intimate relationship between polarity and transport, and showing us why microtubules with their polarity make the best candidate for cellular transport. So that's it for the first lecture of Unit 4. We will finish up this chapter in our next lecture, where we will discuss the third class of cytoskeletal proteins, the actin filaments, and in that conversation we will fully define not only cellular contractions, but muscle contractions, one of the most amazing achievements of the cytoskeleton. Until then, though, we'll just sit pat and go over this material when we meet in class.